Hi guys, so today we talked about DNA replication. So this is a little bit of a cheat sheet on DNA replication and what you'll need to know going forward. So we start with initiation. This is the starting point of DNA replication. It starts at the origin of replication, which forms a replication fork. Helicase then comes in and unwinds the DNA. It breaks the hydrogen bonds between the two strands of DNA, holding the nucleotides together. We have another enzyme that comes in and helps the DNA stay apart. It unwinds it ahead of the replication fork. We get single-stranded binding proteins that come in. These help prevent our two strands of DNA from reconnecting. Then an RNA primer comes in that's generated by DNA primase. The lagging strand has many primers as it is replicated in the opposite direction, and this completes the formation of the replication fork. Then we have elongation. So the DNA code is read on the leading strand continuously from 3' to 5' and gets built in the 5' to 3' direction. The lagging strand gets built in the opposite direction in short segments. These are our Okazaki fragments that start with a primer when then DNA polymerase can come in and bind to those primer sequences and begin adding new base pairing, following our base pairing rules, which work together. DNA primase also will proofread its work to make sure that the correct base pair is added. And then just to note, our base pairs, according to our Chargaff's rule, A binds with T and C binds with G. Once elongation ends, we end in termination. This is where an exonuclease will remove all of the DNA primers or RNA primers, and DNA ligase joins together the Okazaki fragments. At the end of a DNA strand, we have something called a telomere. This is a protective cap at the end of the chromosome that prevents the loss of DNA. We have telomerase that can catalyze the synthesis of the telomere sequences. These don't actually join anything, or they don't code for anything. They're just extra DNA, so we don't lose that DNA. Here, our strands are rewound and consist of one newly formed strand of DNA and one existing strand of DNA. This is where that semi-conservative model comes into place, is that we are saving one strand and making one strand. So we're semi-conserving the DNA strand as we make a new one through initiation, elongation, and termination.